To draw the central dogma is going to be DNA first, then an arrow. On top of that arrow, you're going to say transcription. Underneath, you're going to say RNA polymerase, and a star under that saying enzyme that makes polymers. After the arrow, you're going to say RNA. Then there's going to be another arrow. On top of that arrow, it's going to say translation. Under the arrow, it's going to say ribosome. Next is going to say proteins. And underneath, you're going to say 20 amino acids. Going back to DNA, you're going to draw a loop arrow. On top of the loop arrow, you're going to say replication. And underneath the loop arrow, you're going to say DNA polymerase. To draw a chromosome with exons, first you have to draw the chromosome, and it's going to be one line and then 3 lines through that line on each side that says ORI, a circle, centromere, and then at the end of each of the lines, you're going to put a square, and that's going to be the telomere, and you're going to zoom out to draw the exons. It's going to be a line. You're going to put 2 slashes at each end of the line. Starting from left to right, you're going to draw a circle, which is the enhancer. Then you're going to draw a arrow pointing at the exons, which is going to be the promoter. Then you're going to draw the 4 exons and label them, and the space in between is the introns. After drawing the exons, you're going to draw a square, which is the poly A signal sequence. Mutations. Enhancer. The function is to allow for transcription factors to bind, which calls for RNA polymerase. The mutation, no transcription factors and no active call on RNA polymerase to the promoter. Protein production would then be reduced. Promoter. The function is the initiation site for RNA polymerase to begin transcription. The mutation would mean no transcription start site, which means RNA polymerase doesn't bind and no RNA produced. This means no translation, no protein. Start codon, AUG. The location where ribosome begins translating mRNA sequence into protein. If mutated, the ribosome wouldn't know where to start, so there would be no translation. This would result in no proteins. Stop codon, UAG. The function is that it tells ribosomes where to stop transcribing and codes for release factors so ribosomes disassemble. This mutation means ribosomes cannot stop transcribing at correct sequences, which will make unnecessary amino acids, disrupting protein folding. This would cause non-functional proteins. Exons. The function is that it is protein coding regions of a gene. A mutation is when transcription and translation occurs, but ribosomes will code for incorrect amino acids from the mutated exon. This equals to non-functional proteins. Introns. The function is that non-protein coding regions of a gene that undergoes splicing out before translation. A mutation would be when transcription and translation occurs and the mutation has no effect on the protein. This would create a non-functional proteins. Poly A signal sequence. The function is that it signals poly A polymerase to add chain of adenine nucleotide to 3' end of the gene and prevents degradation by 3' exonuclease. A mutation would mean no poly A polymerase, no production, degradation by exonuclease. This would mean reduced protein production. Consequences of gene mutations outside of coding sequences. Sequence. Effective mutation. Promoter may increase or decrease the rate of transcription. Enhancer may disrupt the ability of gene to be properly regulated. 5-cap UTR/3-cap UTR may alter the ability of the mRNA to be translated in mRNA stability. Splice recognition sequence may alter the ability of mRNA to be spliced properly. Gene reading frames. Coding strand moves from 5-cap to 3-cap, may be 3-cap to 5-cap if the beginning it needs to be flipped for a start codon. Template strand moves in 3-cap to 5-cap. mRNA is coding strand but with a use. Point DNA, or point, and it affects the DNA. Change in a signal base pair, example T to A. The effect is pyrimidine to another pyrimidine, C to T, or purine to another purine, A to G. This is called a transition. Silent. Base sequence altered does not affect polypeptide A. The effect of this is it does not alter the amino acid sequence of the polypeptide, even though the base sequence has changed. Missense affects the protein. Base substitution that leads to a change in the amino acid sequence, glued to val. The effect is that it alters the polypeptide sequence such that the 6th amino acid is changed from glutamic acid to valine. Single amino acid substitution alters the structure and function of hemoglobin proteins. Nonsense affects the protein. A change in the codon that specifies an amino acid to a stop codon in the RNA sequence. The effect of this is that it terminates the translation of the polypeptide earlier than normal, producing a truncated polypeptide. Frame shift affects the DNA. Addition or deletion of a number of nucleotides. This appears as the reading frame is shifted, many codons will change. The effect of this is since codons are read in multiples of 3, this shifts the reading frame. Translation of mRNA results in a completely different amino acid sequence downstream from the mutation. Gene cross. If unlinked, we expect to get a ratio of 1 to 1 to 1 to 1, because the law of independent assortment is a random process. If linked, we would expect to see more parental gametes than the recombinant gametes. This is because recombinants only show up when there is a crossover event within the interval of the genes. The only way we can have an equal amount of recombinants to parentals is if there's a recombination event within the interval between the genes at every meiosis. Distance equals number of recombinants / number of total progeny times 100. Number of recombinants would be the offspring, so the other would be the parentals, so you subtract that, then divide by 2 to split the two parental genotypes. The number of the recombinants will be divided by 2 for the remaining genotypes. Parentals, most, single crossover, next foremost, double crossover, the least. Equation for map. Single crossover plus double crossover divided by the total number of offspring equals answer in centimorgans. Interference equation. Interference equals 1 minus, in parentheses, the first parentheses, number of double crossovers divided by number total progeny divided by smallest distance between genes multiplied by second smallest distance between genes, and close parentheses. Closest number to 1 means that there is high interference. Maternal effect and snail shell twist. Maternal effect. The offspring's phenotype is determined by the mother's genotype, not its own. In snails, the direction of shell twist, right or left-handed coiling, is controlled by maternal genes. The mother's genetic material produces proteins during early development, which determine physical traits like shell twists. Nurse cells in the mother deposit these proteins into the egg, influencing the offspring's phenotype. The offspring's phenotype reflects the maternal genotype, even if the offspring's own genotype differs. Protamines in sperm tightly package maternal DNA, preventing it from influencing early development. Thus, only maternal proteins guide early embryonic development, including shell direction. The maternal effect demonstrates how maternal contributions can dominate early developmental traits. This illustrates the complex relationship between genotype and phenotype and the pivotal role of maternal influences. If a female snail with dextral, right-handed, shell twist produces sinistral, left-handed, offspring, the female's genotype must be heterozygous, little d. Since sinistral shell twist is a recessive trait for the female snail to have an offspring with sinistral shells, she must carry two recessive alleles, little d, even though she shows the dominant dextral phenotype. The tree of that would be female sinistral, little d, male dextral, big D, and the offspring of that would be female sinistral, big D, male sinistral, big D, and that offspring would be dextral, little d, dextral, big D, dextral, big D, and then dextral, big D. X chromosome inactivation, a genetic mosaic. The phenotype of the heterozygous kittens are explained by X inactivation, which ensures dosage compensation in female mammals by silencing one X chromosome, preventing overexpression of X-linked genes. This inactivation is achieved through chromatin modifications, including histone deacetylation by histone deacetylases, which remove acetyl groups, leading to chromatin condensation. The inactivated X chromosome hypercondenses into a bar body, becoming transcriptionally silent. In calico cats, XOXB, this random X inactivation results in patches of cells expressing either the orange XO or black XB allele, producing the characteristic coat pattern. We know that 100% of calico cats are not XY individuals. It must be XX because the calico cat pattern results from X inactivation, which requires two chromosomes. Since the orange XO and black XB alleles are located on the X chromosome, a cat must inherit both alleles XO, XB to express the characteristic patchwork pattern. X inactivation randomly silences one chromosome in each cell, allowing some cells to express the orange allele and others to express the black allele. XY individuals have only one chromosome and therefore can express either orange or black, but not both, making the calico pattern impossible in typical males. Calico coat color is explained by X inactivation, which ensures dosage compensation in female mammals by silencing one chromosome, preventing overexpression of X-linked genes. This inactivation is achieved through chromatin modifications, including histone deacetylation by histone deacetylases, which remove acetyl groups, leading to chromatin condensation. The inactivated X chromosome hypercondenses into a bar body, becoming transcriptionally silent. In calico cats, XO-XB, this random X inactivation results in patches of cells expressing either the orange XO or black XB allele, producing the characteristic coat pattern. The purpose: X chromosome inactivation prevents a double dose of X-linked gene expression in female males who have 2 X chromosomes. Timing occurs during the 16 through 32 cell stage of embryonic development. Random selection: each cell randomly inactivates 1 chromosome to ensure genetic balance. Inheritance: one inactivation occurs, all daughter cells maintain the same inactive chromosome pattern. Results: creates a genetic mosaic. Different cells express genes from different X chromosomes. Example: calico cat fur patterns. The mechanisms controls by X inactivation, XIC. The X gene produces a non-coding RNA that coats the chromosome chosen for inactivation. Exis RNA recruits histone deacetylase enzymes, causing chromatin condensation and gene silencing. Mitochondrial inheritance and its unique characteristics. Mitochondrial inheritance isn't exclusively maternal. Offspring inherit mitochondria only from the mother. Reason: mitochondria in sperm are destroyed after fertilization because they are tagged with ubiquitin for degradation. Only egg mitochondria contribute to the offspring's mitochondrial DNA, in parentheses, mtDNA. This inheritance pattern does not follow Mendelian laws, segregation, or independent assortment. Mitochondria doesn't follow law of independent assortment because, unlike chromosomes that neatly align on the metaphase plate during cell division, mitochondria are randomly distributed throughout the cytoplasm. So when the cell divides, they end up in whichever daughter cell they happen to be closest to. There's no organized system for the distribution like there is for chromosomes. Mitochondria doesn't follow the law of segregation because the law of segregation suggests that every daughter cell gets equal amount of mitochondria compared to every other daughter cell. However, mitochondria are not distributed equally to both daughter cells after division. Whether they are in the cell when division happens is what determines where they go. Mitochondrial DNA is inherited uniparently from one parent only. 2 key terms: heteroplasmy, mixture of normal and mutant mitochondria within a cell. Homoplasmy, all mitochondria in a cell are genetically identical. Disease manifestation: a threshold level of mutant mitochondria must be reached for symptoms to appear. Severity increases with a higher proportion of mutant mitochondria equals less ATP production equals cellular dysfunction or death. Importance: understanding mitochondrial inheritance helps explain how non-Mendelian genetic diseases develops and how mutations affect cell function. The difference between normal mitochondria and mutant mitochondria is that mutant mitochondria do not produce ATP in sufficient quantities for optimal function. Epigenetic modifications and DNA accessibility. The initiation of transcription can be controlled by the state of the chromatin at the gene because the initiation of transcription is strongly influenced by the chromatin state at a gene. When chromatin is loosely packed, euchromatin, DNA is more accessible to transcription factors and RNA polymerase, allowing transcription to begin. In contrast, tightly packed chromatin, heterochromatin, restricts access, preventing initiation, acetylation, and DNA expression. Histones are proteins around which DNA is wrapped. Acetylation of histones relaxes the chromatin structure, allowing transcription factors easier access to DNA. This is carried out by histone acetyltransferases, aka HATs, which add acetyl groups to histones. Acetyl groups neutralize the histone's positive charge, reducing their attraction to negatively charged DNA. Genes in acetylated regions are more likely to be actively expressed. Deacetylation. Histone deacetylases, HDACs, remove acetyl groups. This causes chromatin to become more condensed, restricting transcription factor access. Deacetylation leads to gene repression. Methylation and gene silencing. DNA methylation occurs at cytosine bases with CpG dinucleotides, characterized by DNA methyltransferases. Methylation compacts chromatin and prevents transcription factor binding, silencing genes. DNA methylation typically affects gene expression by silencing genes by compacting chromatin. The relationship between methylation and transcription factors is methylation interferes with transcription factor binding by contributing to silencing. Methylated DNA regions are generally associated with gene repression. Biological impact. DNA methylation can cause long-term silencing, plays roles in development, cellular differentiation, and disease. The interplay between acetylation, deacetylation, and methylation controls chromatin structure and gene activity. Understanding genetic imprinting. Definition. Genetic imprinting is when gene expression depends on the parent of origin, whether the gene is inherited from the mother or father. It's a form of sex-dependent inheritance involving selective silencing of one parental allele. Example, IGF2 gene. IGF2, insulin-like growth factor 2, is expressed only from the parental allele. The maternal allele is silenced. This selective expression is essential for normal fetal growth and development. Mechanism. Imprinting is controlled by epigenetic marks, especially DNA methylation. In the paternal allele, methyl groups mark the imprinting control region, ICR, activating IGF2. In the maternal allele, CF proteins bind to the ICR instead of methyl groups silencing IGF2. These marks ensure that only one allele is active, preventing overexpression. Germline reprogramming. In males, silencing imprints are removed. All sperm carry active IGF2 alleles. In females, imprints are removed and then re-added. All eggs carry silenced IGF2 alleles. IGF2 H19 locus. IGF2 and H19 share an enhancer. The ICR determines which gene is transcribed. If methylated, males, enhancer activates IGF2. If IGF2 and H19 share an enhancer, when the imprinting control region is methylated, the shared enhancer directs RNA polymerase to the IGF2 gene. If CTCF bound females, enhancers activates H19. Methylation can affect more than promoter/enhancer blocking. It can redirect transcriptional activity. Medical relevance. Abnormal imprinting can cause diseases like Prader-Willi syndrome and Angelman syndrome. The orders of process of imprinting in males for the IGF2 gene 1. Any individual has an active copy of the IGF-2 from the father and an inactive copy from the mother. And 2. In the germline cells, CTCF proteins are removed from the imprinting control region. 3. All chromosomes in the germline cells now carry active IGF-2 genes. 4. Meiosis occurs and now all gametes carry active IGF-2 genes. 5. When one of these genes merge with an egg cell, the gametes will transmit its active IGF-2 gene to the offspring. The order of the process of imprinting in females for the IGF-2 gene. 1. For most mammals, every individual inherits 2 copies of IGF-2, an active one from the father and an inactive one from the mother. 2. In germline cells, all CTCF proteins are removed from all chromosomes. 3. New CTCF proteins are added to all the chromosomes in the germline cells, inactivating all of them. 4. Meiosis occurs and all the resulting gametes carry only inactivated CTCF-bound chromosomes. 5. When this egg cell is fertilized by a sperm cell, the egg cell will always carry inactivated IGF-2 genes, and the sperm will give the active IGF-2 gene for the offspring. For IGF-2, offspring will inherit the active gene only from the father. This does not mean the offspring will have the same phenotype as the father, because when the father makes gametes, all of the silencing imprints are removed. This means that even the gene he got from his mother will be active in the resulting gametes, which could be passed to the offspring.